Okay, welcome back. And now we're going to talk about microscopy and some of the common components of cells and why cells are small in this lecture. Okay, so the two types of microscopy that we will talk about in this class and that I want you to be able to recognize images of is the light microscopy, and this is the type you've used in school before. So uses light to illuminate whatever you have on your slide under here. Pretty simple, basic type of microscope. It's also called bright field, so you may hear either term. And remember that the whole reason we use microscopes is because cells are so small, we can't see them very well without a microscope. So I want you to understand these two different types of microscopes and what you can actually see with them and what you can't. The second type we'll talk about is the electron microscope, and it's this amazingly huge, complex microscope that actually uses electrons to create the image versus light to create the image. And we're going to talk about two different types of electron microscopy, transmission and scanning. Note that only with light microscopy can you look at live cells. Everything that you see under an electron microscope will be dead. We have to fix the cells so that we can shoot those electrons through them to get the image. Okay, so light microscopy. You can see cells, but only really a few organelles, nothing much smaller. So you've seen this picture already here where we're looking at a cheek cell, and you can see the little bacterial cells on it. And the organelle you can see here is you can see this dark staining that represents the nucleus. Okay. This is a dead cell because it has been stained. So if you, oh, that's weird. Okay. I never get phone calls. There we go. Okay. Because it has been stained, the stain kills the cells. We have to fix the cells. Okay. So here are some bacteria that have also been stained. So note that most cells are see-through. They don't have color to them. This is a lovely paramecium. This is a protist, single-celled organism, and you can kind of see, you can kind of like look right through it. It's see-through. So a lot of times we have to fix the cells and stain them so that we can really see them well under the microscope. These would be live plant cells, and all these green little circles, those are your chloroplasts. We'll talk about chloroplasts in a little bit. And these are naturally green, so they give the plant color that you see on the trees and bushes. And you can also see these organelles excuse me, under the light microscope. So a very few organelles you can actually distinguish under the microscope. In the paramecium, it has some food vacuoles and things that you can kind of see. We'll look at this a little bit more, but it also has cilia. But really, with light microscopy, you're looking at cells, you're looking at cell shape. You can stain them, and they'd be dead cells, and you can see a little bit more detail, but you're not going to be able to see the ER, the Golgi, some of the other organelles that we're going to talk about. Again, this is just showing you, here's an amoeba in its natural beauty, and here is a stained amoeba. So these would be dead. They're stained pink. Amoebas aren't naturally pink. So with light microscopy, you can see both live and dead. If you want to see what cells look like live moving, you can check out this video. I put super long but super cool, so obviously you don't have to watch it all, but it's pretty fun to watch different cells move around. So if you're wondering why I look over here, I'm checking my notes to make sure I've covered everything I want to cover for each slide. A type of microscopy that we're not going to really talk about, but you've probably seen images of it, is the use of fluorescence. So there are proteins that fluoresce naturally, a protein called green fluorescent protein. We can also use antibody staining for fluorescence. So some of these amazing images, these are all dead cells. To stain them, we have to kill them, unless they have GFP, and now I'm making things more complicated. But I just wanted to tell you, if you see these types of images, this is fluorescence, this is with another special type of microscope, and if you take cell biology with me, you'll learn more about fluorescence. Okay, and then the electron microscope, this is giving us these amazing details. Okay? So we have transmission electron microscopy, and what's happening is you're seeing internal structures. The electrons are transmitting right on through the cell. So with TEM, it's really a 2D, like a flat image. It's the what do I want to say? The internal structures. And I should write this word, transmission. Okay, so it's just a nice, think of it as a flat view, but so many details. I mean, look down here. Now you see the nucleus, the nucleolus. You can clearly see mitochondria, vacuoles. These two right here are what we call false colored, because electron microscopy, electron micrographs, so micrograph means the image you get from microscopy, they're black and white, okay? And then you can, with the computer, color them. So that's why we call these false colored. Scanning, SEM, S stands for scanning electron microscopy. And this is where you get a 3D image. So this is scanning across the surface. So transmission, the electrons are going through, they're transmitted through. Scanning, the electrons go across the surface. So you get these really cool looking pictures. So like these are red blood cells and some macrophage and some fibrin in a blood clot. This is a chloroplast. So we're still seeing internal structures, but you can see how it's like 3D. Like you can kind of you can kind of envision the thickness and the depth. Okay, so a scanning is going to give you a 3D surface image. Transmission is going to give you this 2D flat, but it's all about details. This is a much greater magnification. So let's make sure you understand the difference between magnification and resolution. So magnification, as it says here, is how much bigger a sample appears than what it truly is. So we've magnified onto this dog's nose. Resolution is really about the detail. So can you distinguish between two spots, okay, two images? So you can see that you have much better resolution on the left-hand side. It's all blurry over here. So this would have lower resolution. They're both images at the same magnification. It's just which camera actually has better optics to resolve or to clarify these different what am I trying to say? I think you know what I'm trying to say. Which cameras or microscopes can have higher resolution, higher ability to look at objects and be able to tell that they're distinct. Okay. Good. So one more part I want to talk about in this video is what makes a cell, okay? And why cells are small. And then we will pause, and then we will go on to organelles of the cell. Okay, so all cells have 4 essential components. And those 4 essential components are a cell membrane, okay? So you might also see this called a plasma membrane. Same thing. It's basically the border of the cell. So prokaryotic cells have cell membranes. Eukaryotic cells, both animals, plants, fungi, protists, all have cell membranes. All cells have something called cytoplasm, and we're going to define this in the next video. But cytoplasm is the fluid inside all the cells. All cells have DNA as the genetic material. And we talked about this in the last video, how some viruses don't have DNA as their genetic material. Viruses don't have a cytoplasm. Some viruses do have a membrane, we call it an envelope, wrapped around it, but they steal it from the cell they've infected. The last thing that all cells have, this is our 4th, is ribosomes. And again, we're going to talk about those in a minute, but they are for protein synthesis. So, 4 components for all kinds of cells: cytoplasm, DNA, ribosomes, cell membrane. This allows the cells to have their own genetic information and to be able to produce the proteins that they need. Okay, last topic for this video is, why are cells small? So if you compared a little chihuahua to a big elephant, okay, they have the same size cells. Elephants just have more cells because they're a bigger organism. Cells have to be small because of something called diffusion. And I got this image from this website here, and if you scroll down to the bottom, there's a good little video about diffusion. Diffusion is just the passive movement of molecules from high to low concentration. And we'll actually talk a lot about diffusion, I think in the next chapter. So molecules have to be able to get from the surface of a cell through the plasma membrane to wherever they need to go in the cell, and waste molecules need to get from the cell out. Okay? And these don't have little motors to move them around. It's all movement by diffusion. So if you have a small cell, the molecules can diffuse and they don't have a long way to go. As you increase the size of a cell, you can see that the molecules got about the same distance, but they never made it all the way to the inside. And that would mean that if this was a real cell, that cell would be kind of starving in there, or it might have a lot of waste to build up. So cells have to be small because of diffusion, because of this passive movement of molecules. The concept that we want to talk about is this surface to volume ratio. And what you want is you want a large surface area, so think your plasma membrane, to small volume ratio. So you want a large surface area, you want a small volume. And so if cells are really big, like this, they're, oh, I didn't put the ratio on here. Well, they have less surface, so the molecules can only come in here and here and here, and the volume is so big that the cell wouldn't survive. So what's better is to have a whole bunch of small cells which have a larger surface area to volume ratio. Now, you might be thinking, let's see if I can go back. Okay, look at the big size difference between prokaryotic cells and eukaryotic cells. How can a eukaryotic cell work if it's so big compared to a prokaryotic? And that's because eukaryotic cells have organelles. So all of these organelles here in this paramecium, and there's a whole bunch that you can't see, they take up space, and so molecules can get to them, and then sometimes the organelles can take them where they need to be. So you can have a larger cell if you have organelles. Those help with the task, those make the cell more efficient. So that's why a eukaryotic cell that looks so much bigger than a prokaryotic cell still functions. But the concept of why are cells small is because of diffusion, and therefore you want a large surface area, small volume. Okay, so next we're on to parts of a cell. You will have a sheet like this. I encourage you to fill it out maybe in a day or so and test yourself with this. This is a great way to study. So we're going to stop here, and the next lecture video will be about eukaryotic organelles.